Hi all. Um, I am Noor. I'm a graduate student at the University of Toronto, and today I'll be talking about associating ion channel alternate splicing with neuronal intrinsic electrophysiological properties using Patch-seq. So, what is alternate splicing and why is it important? Alternate splicing is a cellular process in which exons from the same gene are joined in different combinations, leading to different but related mRNA transcripts. It is important because almost all mammalian multi-exon genes undergo alternative splicing. It is especially important in the context of central nervous system because it's highly conserved in mammalian nervous tissues and it has been shown to be implicated in multiple neurological disorders. Previous studies have shown that alternate splicing in ion channel genes regulate intrinsic electrophysiological properties. And in this particular example, this gene has two alternatively spliced isoforms. And for the isoform on the left, the blue exon is excluded. And for the isoform on the right, the blue exon is included. And this exclusion and inclusion of this blue exon um, causes give rise to two different ion channel isoforms that have different biophysical properties. And when these two ion channel isoforms are expressed in the neurons, they give rise to different electrophysiological properties. For this project, we used the Patch-seq dataset collected by the Allen Institute for Brain Science. So this dataset contains data from more than 4,000 GABAergic interneurons from the primary visual cortex of adult mouse. So what's special about this dataset is that it contains both intrinsic electrophysiological properties and also transcriptomic data. And this gives us a unique opportunity to correlate splicing events extracted from the transcriptomic data with the electrophysiological properties. And for this project, I adapted a method called S-seq. S-seq is a method developed for detecting differentially splicing events between cell types from single-cell RNA-seq data. And you can see from the heat map on the right, each column is a cell type, and this PSI value is just a measure of splicing. And you can see that for each alternative splicing events, which are shown in the rows, it has very different values across different cell types. But for the same cell types, the value of free expression is relatively stable. The S-seq method uses a statistical model called the Dirichlet multinomial generalized linear model. And in this model, the response variable is a vector of splice junction counts in group G conditioned on the total number of splice junction counts in group G. And the predictor, as shown here, X is a binary variable. And this linear predictor represents the underlying splice junction proportions. So in the original S-seq method, the predictor is a binary variable that represents the cell type label. It is 1 when the cell is included in the particular cell type and is 0 when it's not included in the cell type. So to make this method work with electrophysiological properties, which are continuous variables, I extended the S-seq method to make it include cases where the predictors are continuous variables. And this figure shows you the overall workflow of the project. So I extracted electrophysiological properties from the Patch-seq dataset using a package called IPFX, which is a standard set of tools for this kind of task. And then I also extracted, I counted the splice junction reads from the transcriptome data in the Patch-seq dataset. So the results, so we conducted likelihood ratio tests for each pair of splicing event and electrophysiological properties, and this heat map shows you the negative log 10 p-value of the likelihood ratio test, and the asterisk means that this association is significant, and we can see that real base upstroke downstroke ratio and action potential half-width are the electrophysiological properties that have the most number of splicing events that are correlated with them. And this is results from the gene site enrichment analysis. And you can see that genes containing differentially spliced junctions are enriched for ion channel genes. For example, ion transmembrane transporter activity, voltage-gated channel activity. And the advantage of using the Patch-seq method is that it allows you to detect splicing events that are novel. So these events contain splice sites that are not annotated in the reference genome. So you can see that actually a large fraction of the splicing events that are associated with electrophysiological properties are novel. So in conclusion, we found that genes containing electrophysiological property-associated splice sites are enriched for ion channel genes. And using the electrophysiological properties as predictors of the generalized linear model identifies more differential splice site usage than using the subclass labels as predictors. And a large fraction of the if-it's associated splice sites are novel, and many if-it's associated splice sites detected by the Dirichlet multinomial GLM have been studied in the literature. Thank you.